Hello, good morning to all of you. Myself, Dr. Suman Rawat from Department of Biosciences, Himachal Pradesh University. Students, today we will discuss the general characteristics of cryptogams and differences among them. So, first of all, we will start with the classification. Kingdom Plantae is divided into two major categories. In first category, plant body is not well differentiated. These kind of plants come under the category of Thallophyta, for example, algae and fungi. In the second category, the plant body is well differentiated, but it may or may not contain the vascular tissue. If they don't possess vascular tissue, they come in the category of Bryophyta, and if they have the vascular tissue, they may be pteridophytes, gymnosperms or angiosperms. So further, they are divided into two other categories which do not produce seeds are called as pteridophyta, and which produce seed, and if they are naked then the plants are called as gymnosperms, and if seeds are enclosed inside the fruits they are called as angiosperm. So kingdom Plantae is divided into five major categories. They are Thallophyta, Bryophyta, Pteridophyta, gymnosperms and angiosperm. Let us discuss the general characteristics of bryophytas. The division Bryophyta include over 25,000 species of non-vascular embryophytes such as mosses, liverbirds and hornbirds. They are small plants. Height ranges from 2 cm to 60 cm and generally grow in moist, shady places. They don't attain great height because of absence of roots, vascular tissues, mechanical tissues and cuticle, and mostly they are terrestrial but require external water to complete their life cycle. Hence they are called as the amphibian of plant kingdom and also known as Atracheata or Atracheophyta because they lack the vascular tissues. They have heterologous haplodiploidic type of life cycle. The main plant body is gametophytic on which the sporophyte is dependent for the nutrition. The thalloid gametophyte differentiate into rhizoids, axes and leaves. Vascular tissues are absent and the gametophytes bear multicellular and ejected sex organ. The sex organs are antheridia and archegonia, and sexual reproduction is oogamous type, in which male is motile and female is non-motile. Multicellular embryo dwells inside the archegonia. So bryophytes are first in which the embryo is formed. In Thallophyta, embryo is never formed. Sporophyte is differentiated into foot, seta and capsule. Foot is for the anchoring of the nutrition from the gametophyte and seta for the conduction, while capsule enclosed the haploid spores and elaters, and elaters have the spiral thickening which helps in the digestion of capsule. Capsule produces haploid myospores of similar type. So in bryophytas, spores are homosporous. These spores germinate into the juvenile gametophyte and called as protonema. So protonema is a thread-like chain of cells that forms the earliest stage of development of the gametophyte in the haploid phase in the life cycle of mosses. In bryophytes, sporophytes is dependent on the gametophytes. This is an important character of bryophyta. Let us discuss similarity between the bryophyta and pteridophyta. Both are terrestrial, both have heteromorphic type of alternation of generation, both have multicellular and ejected sex organ. Asexual reproduction is carried out by the means of mitospores and accessory spores are absent. Sexual reproduction is oogamous type where the male is motile and female is non-motile, and both have the necessity of water for the reproduction. These are some common names for both Bryophyta and Pteridophyta. They are Archegoniate, Embryophyta, and Cryptogams, because both have the sex organ Archegonium. So they are also called as the Archegoniates. Both have the embryo, so they are also called as come in the category of Embryophyta. In algae and fungi, there is no embryo, while in Bryophyta, Pteridophyta, Gymnosperm and Angiosperm all have embryo in their life cycle. They are also called as the cryptogamous plant because we can't see the reproductive structure in algae, bryophytes, and in pteridophytes. Gymnosperm and angiosperms, they are called as the phanerogams because we can see the reproductive structure in them. Let us discuss the habit and habitat of bryophytes. So bryophytes are the simplest terrestrial plant which lack complex tissue system, and they are 2 to 5 centimeter tall, others less than the 10 centimeter long. They commonly grow in damp, humid and shaded localities, widely distributed throughout the world, and also called as the amphibian of plant kingdom because they can live in soil but are dependent upon water for the sexual reproduction. The main plant body in the Bryophyta is haploid, as the main phase is gametophytic. It produces gametes, and sex organ in bryophytes are multicellular. Male sex organ is called as antheridium, which produces antheriozoids, while the female sex organ is archegonia, which produces egg. Antheriozoids are released into water and come in contact with archegonia. Egg and antheriozoids are combined to form the zygote, which is diploid in number 2n. Further, zygotes develop into the sporophyte. In the Bryophyta, sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some sporophytes undergo meiosis, a reductional cell division to produce the haploid spores, and these spores germinate to produce the gametophytes. This is the haplodiplontic life cycle of the bryophytes. So let us discuss the differences between the pteridophytes and the bryophytes. So bryophytes, a name derived from the bryon, that means mosses, while the pteridophyta name derived from the pteron, that means feather. They are called as the amphibian of plant kingdom, Atracheophyta, because the vascular tissues are absent, while the Pteridophyta, they are known as botanical snake, Tracheophyta, and also vascular cryptogams. Bryophytas are the first land plant, while they are first rooter plant, spore producing vascular plant, lower vascular plant, and also known as the first vertebrates of the plant kingdom. Size of the cell in case of Bryophyta is small, and tissue required for the growth are absent. Apical growth is absent, conductive tissues are absent, secondary growth is also absent, while in the Pteridophyta cell size is large and only in two genera, that is Batrachum and Isotis, show a slight secondary growth. Tissue differentiation in bryophytes is very little. Even cuticle is absent and life cycle is haplodiplontic. That is, the main phase is the haploid, that is the gametophytic. While in case of the Pteridophyta, there is tissue differentiation, and in two species, that is the Alsophila and Scythia, which is known as the tree ferns, they can reach up to the 25 meter in height. Cuticle is present in Pteridophyta, even it is present in Gymnosperm and Angiosperm also. Life cycle is diplohaplontic, where the main phase is diploid and main plant body is sporophytic. First diagram is of Bryophyta and second diagram is of Pteridophyta. In the first diagram, the life cycle is haplodiploidic. In the second where the sporophytic phase is the main dominating phase, and life cycle is diplohaplontic in case of Tradophyta. The dominating phase in the Bryophyta, it is gametophytic. So gametophytic is independent and it is autophytic. It may be leafy in case of mosses, and in liverwort it may be leafy or thalloid, while in hornwort it is thalloid. 80% of the Bryophytas are leafy gametophyte, while in the Tradophyta dominating phase is sporophytic, and the gametophyte never will leafy, and maybe it is thalloid or filamentous type. In Bryophyta, sporophyta dependent upon the gametophyte and not differentiated into root, stem and leaves, but differentiated into foot, seta and capsule, and it is short-lived and less complex. Roots are absent. Rhizoids are present in the Bryophyta, while in the Tradophyta, sporophytes is independent and it is differentiated into root, stem and leaves, and it is long-lived and complex, and roots are present. In Bryophyta, stem is absent, but it is seen in the mosses, and it is not of true type. It is called as the coil. Even leaves are also not true life. It is without the vascular stand, without the stomata, and it is haploid, and they are called as the phyllid or phylloids. Photosynthesis is carried out by the whole body, and vascular tissues are absent, while in the Tradophyta stem is present and it is with the apical growth. True leaves are present and only leaves are photosynthetic, and vascular tissues are present. That is why they are known as the vascular cryptogams. Asexual reproduction in the Bryophyta is non-sporangiate, and they are strictly homosporous. All spores are same, while in the Tradophyta, asexual reproduction is polysporangiate, and majority of the members are homosporous, while 9 genera are heterosporous. Examples are Selaginella, Isotis, Stylites, Marshallia, Pilularia, Regnalidium, Salvinia, Azolla and the Platysoma. So character heterospory is first time seen in the Tradophyta. The adoption of heterospory and the retention and germination of a single megaspore within the megasporangium to form a female gametophyte led to the phenomenon of seed habit. A characteristic feature of the spermatophytes. A seed is that ovule which contain an embryo developed as the result of fertilization. So this is an important character of the Tradophyta. Vegetative reproduction is very common in Bryophyta. Archegonia in liverwort and mosses have the 6 rows of neck cells, exception only in Borrelia where only 5 rows are present. Neck canal cell 8 in case of liverworts, 10 in case of mosses, 4 to 6 in case of hornworts. Ventral wall, it is single in liverworts and double in mosses and absent in hornworts, while in the Tradophyta, vegetative reproduction is less prevalent and archegonia is less massive, or you can say that it is less developed. In Selaginella, only 1 neck canal cell and 1 ventral canal cell is present. Exception is only in Dryopteris where the 4 rows of neck is present. Spermatozoites and entheritiozoites are biflagellates in Bryophytas. Mucilase, sugar, protein, inorganic salt produced by absorbing the water by the neck canal cell, and that attracts sperm. This type of attraction is called as the chemotaxic, while in the Tradophyta, entheritiozoites are biflagellate in case of Selaginella, and they are multiflagellate in ferns. Chemicals are absent, but compound like citric acid or their salt are seen in Lycopodium. Malic acid or fumaric acid are seen in Selaginella, which plays an important role in reproduction. Bryophytes are primitive type of plants, and the pteridophytes are the advanced types of plants. So this is all about the lecture. Thank you so much.